This is a question one of my PhD students brought to me yesterday. I think this is from an undergraduate statistics class. The question says, the least squares estimate of the slope, it's about a simple linear regression. It's not exactly equal to the true value of the slope, or at least there is a very large probability that these two are not equal. Which of the following helps us measure how far away an estimate of the slope might be from the true value? There are 5 answers: the t statistic, the correlation coefficient, the p-value, the mean of the least squares estimator of the slope, the variance or standard deviation of the least squares estimator of the slope. So what do you think? This is related to what we talked about, the frequentist statistic in our first 2 lectures.
Should be E, right?
Yeah, actually, yes, the answer is down here. So the answer is put here, so you got it. But my student just asked me, why isn't it A? Why isn't it the T statistic? You know, when we do regression, if you do that in R, say using the linear function for Lm, that's the linear function in R, you would actually get a p-value associated with a T statistic for the coefficient. And it is about whether you want to reject the null hypothesis that the slope coefficient is 0.
I mean, the t statistic, isn't that related to whether the slope is 0?
Exactly. It's about the null hypothesis, whether you want to test the hypothesis you want to test against. The null hypothesis is slope is 0. Although the t statistic has a denominator in it, and that denominator is trying to capture the standard error of your slope estimate, but it's not really. T statistic itself is not an uncertainty quantity. It's not trying to quantify the uncertainty of your slope estimate. In fact, that quantity for uncertainty is the variance. So that's why in frequentist statistic, whenever we have an estimator for a parameter, we want to talk about the variance of that estimator, because that will give us some idea about how largely would that estimator value change as we repeat our sampling. So it's capturing the sampling variation, and that's the core of frequentist statistic. Like we said, in frequentist, we assume the parameter to be fixed but unknown, but we assume the estimator, which is a function of our data, to be random. As we repeat our sampling, we get different data and we get different values for that estimator. That's the core. So do we have questions about this question before we go into the main topic of our class? Okay, so I guess we're good. So let me switch my screen to our lecture. And also, by the way, I have posted the discussion about the robustness of t-test, 2-sample t-test to our CSLE. So if you're interested, you can check it out. There is some very interesting discussion about when the t-test can be used when the data are not normally distributed. Okay, so let's, oops, I'm just sharing this again. Okay. So now let's move back to our lecture. Last time we started talking about normalization. Okay, normalization is something that is very important for our data analysis because it is essential to make sure our observations in different biological samples, let's say a biological sample may be a repeated experiment. So in this case, if you want to compare the same genes measurements in different samples, you want to first make sure the samples are comparable with each other. Otherwise, the values are not meaningful for comparison. So last time we mentioned that for RNA sequencing data, what you first need to do is for every gene, you need to normalize the number of reads in that gene by the sample's sequencing depth. Since the sequencing depth is just the amount of information you extract from sequencing machine, and your gene expression rate count is linearly correlated with the sample's sequencing depth. So that's how we do it. But there are two units that are widely used in the literature. One is RPKM or FPKM. They are very similar to each other. The other is TPM. So the difference between RPKM and TPM is whether you want to adjust for, whether you define the sequencing depth as the total number of reads or the total number of RNA molecules. So in TPM, the unit, the sequencing depth is based on the number of molecules. So that's why for every gene, we divide its number of reads by its length so that we can make the different genes to be comparable since their length is adjusted. Okay, so, and in our last lecture, if you check the recording, I posted several blogs and research papers about this topic. And also on our CCLE, I posted the first paper, I think it was from 2012, that introduced the concept TPM. Even though it was not published in the journal I'm familiar with, but I'm very surprised, I'm very happy to see that it has received very significant attention from the field, and many people agree with its analysis and now switch to TPM for many of its analysis. So I would say if you are doing analysis now, probably I would go for TPM because I think it's more justified. Okay, so a lot of times, once we do this, right, our data will be in TPM unit. So every gene will have a TPM value in a sample, but that's often not enough. So there may be other batch effects we do not adjust yet. So then there are some other approaches for adjusting batch effects. So I'm just going to mention a few here for your information. So the second thing people may consider, okay, probably I should say that this is the first type, adjusting for sequencing depth. Adjustment for seq depth. And the second type is to adjust for other unknown batch effects based on some genes which you know should not vary across samples. So this is adjustment based on so-called invariant genes. So in biology, the most famous invariant genes, I would say, are housekeeping genes. So these are the genes that must be expressed in any biological sample, and you expect that their expression levels to be pretty constant across samples. If that's what you believe, then you can identify those genes in every sample and make sure that their expression is similar in every sample. So what you could do is that, let's say, maybe I can say that you have housekeeping genes. Okay, so probably I can say that I have a set. Okay, I have a set H, and this set H is a subset of all my genes. Let's say I have P genes in total. Okay, this is a subset of it. And I first need to identify in some way. Then what I could do is that I could calculate a factor by compare for every sample based on its set. Okay, this is the set of invariant genes. And now let's say that for my sample I could just take the summation for its genes in the set. Okay, so let me call this Xij. And if the J is in the set, I would take the summation. So Xij, by the way, is the expression level of gene J in sample I. So I would say here, probably my Xij is already TPM. So after those pre-processing, I start from there. Okay, so then I would expect that this sum should be quite constant across sample I. So therefore, what I could do is that I could calculate a weight factor, or I would say maybe a scale factor, probably that's better, a scale factor, scaling factor for every sample I.
Dr. Lee?
So, yes.
Is this set of invariant genes, tissue or cell type specific in practice?
I would say it depends on whether your samples are replicates of one condition, one tissue, or they are different tissue samples. So if your samples are from different tissues, then this invariant gene set will be the genes whose expression do not change across those tissues. So in other words, the invariant gene set definition will depend on the samples of your analysis.
And do you know how they initially determine these genes? It seems like a really hard problem.
Yeah, so I will talk about that very soon after we said this. Yeah. Okay, so assuming that we have this set known, and then given this sum, we can define a scaling factor for sample I. Just as, let me call that WI, it would just be, probably I can give this a notation, this total, I can call it SI. It would just be the SI over, you can do it the mean of, let's say, S1 to Si or Sn. Suppose that you have n samples, okay? I from 1 to n. You have n samples in total. So that will give you a scaling factor. So you can imagine what it will be like. If this Wi is much greater than 1, it means that those invariant genes are having a higher than expected level in sample I. And so therefore, the sample I should be adjusted to be lower values. On the other hand, if Wi is less than 1, then you may want to scale up sample I. So therefore, based on this definition, then the adjusted expression level would just be equal to Xij divided by Wi. So you can see that is making sense because when Wi is greater than 1, you divide it, then you will make the values smaller. If Wi is less than 1, you divide it, you will make the value greater. So that would be the adjusted value. And we can call this Xij prime for the new value. Okay, then back to Gabe's question about how do we define invariant gene set. So I actually found one reference, which was a very famous paper. It's called dChip, which was a very famous paper for microarray data analysis. It's 2001. Even though microarray data have different scales and units from the RNA-seq data, but many ideas can be carried over because they're both gene expression measurements. So for that, let's take a quick look at this paper. Okay, so let me switch my screen to that dChip paper for now. And we can read what is written there together. Okay, so this is the dChip paper, and this is a book chapter which I will post to CCLE. So there is a normalization step. So specifically, we can go down to this paragraph. It says we use, maybe I can make this bigger. We use an iterative procedure to identify a set of probes. So you can think of the probes as our genes here, because in microarray data, we use a probe to capture a gene, which presumably consists of points from non-differentially expressed genes. Specifically, we start with points of all probes. If a point's proportional rank difference, so this is defined as the absolute rank difference in the two arrays divided by the total, is small enough, this is kept for the new set. By small enough, we mean less than this small number or another number, but this is arbitrary thresholds, which we can adjust for. So these parameters were chosen empirically to make the selected points in the invariant set thin enough to naturally determine a normalization relation. In this way, we may obtain a new set of points, and the same procedure is applied to the new set iteratively until the number of points in the new set no longer decreases. A piecewise linear running median curve is then calculated and used as the normalization curve. After normalization, the two arrays have similar overall brightness. Okay, so what does it mean? So here, the normalization procedure in dChip is slightly different from what I talk about. I basically just use linear scaling for normalization. But what they did is that they are doing a curve, right? So essentially, probably we can see here. So a normalization relation can be understood as a curve in the scatter plot of two arrays with the baseline array drawn from the y-axis and the array to be normalized on the x-axis. So in our context, the y-axis is one sample, the x-axis is another sample. A line running through the origin is the multiplicative normalization method. So you can imagine that if the data are perfectly corresponding to one another, the data points should lie in the diagonal line, y equals x, between the two samples. But if not, you may want to adjust them in some way to that line. So for that step, there could be multiple approaches. So for example, one thing you could do is that you could run a nonlinear regression. For example, in statistics, a very famous tool for doing that is called a smoothing spline. So which means that maybe I can show that to you very soon, is to try to fit a line through your data points. And then you will adjust the value based on that regression line. Okay, so let me switch back to my handwriting. Okay. So regression-based adjustment is like this. So this is what I called linear scaling. It's the simplest way for doing normalization. Or you could do regression-based normalization. So what does that mean? So this means that if you have a sample to be aligned into, so let's say this is the ideal sample or target sample you want the other sample to be like. And this is the sample to be normalized. Okay, that's the case. And then every dot here, every dot is a gene. So this is, for example, gene I. So like I said, if the two samples agree perfectly with one another, the dots should lie perfectly in the diagonal line. But let's see that the data does not look like that. For example, the data looks like this. And our diagonal line is this. Okay, then clearly for those genes that are highly expressed in this sample, they should be actually, so basically these samples are to be normalized here. They are more highly expressed in my current sample than the target or ideal sample. So then how do I do that? So one way to do that is to fit a regression line, which means that you fit a line through these points. Okay, for example, we can say this is probably a smoothing spline or other non-parametric regression. And once you do that, you can just convert every point, let's call this f hat, okay? Then if I call this as my x, so this is gene I, okay? This is my xi, and this is gene j. I call this xj. Then what I could do is that I just convert xj to f hat xj. So doing this adjustment can make the sample in the x-axis to be more like the sample in the y-axis. So that's what we call the regression-based adjustment. And in that iterative procedure, what does it mean? It means that it first try to find a rank for every gene in the two samples. So here, maybe I can say that, so for gene I, okay, so we will have a rank 1 and rank 2 in the two samples. Let's say that I rank the gene from the lowest to the highest, or highest to the lowest. Maybe that's better to understand. So let's say this gene is ranked 20 in sample 1 and ranked 25 in sample 2. And let's say that we have the rank that go from, say, 1 to 20,000. That's the total number of genes in human, right? So then you see that this gene actually has very similar rank in the two samples. So then in that iterative procedure, we try to identify this gene as an invariant gene. So we will put that gene into the invariant set. So this looks good. So then this gene will go to the set H. But let's say if I have another gene K, whose rank in the first sample is 30, but whose rank in the second sample is 4,000, then this gene will not go to that set, okay? So we're basically trying to identify the genes based on their relative rank agreement, and that will determine the gene H. And this is based on the gene set H to draw this line. So with this, because we believe these genes should lie in the diagonal, and after we fit this line, we will apply them to all the genes to do the adjustment. And after we do the adjustment, the rank may be changed. So then we will redo the rank and redo the calculation and update the H. So that's the iterative procedure over there. But in the linear scaling I talk about, you can see that the change I make to the values will not change the rank, because the low expressed genes after the scaling in that sample will stay as low expressed. The highly expressed will stay as highly expressed. So the way I do, there's no iteration at all. It's just based on the set H to start with, and we're done. But based on what they do, if you have done some nonlinear scaling, nonlinear regression like those, since you're fitting this line just based on set H, and you apply it to other genes, it's possible that some other genes' rank will be changed. Does that make sense?
Professor Levy, just one question. How did they get the target ideals? What is the y-axis again?
I'm sorry, who is speaking?
Harold is there.
Sorry, okay.
Hi.
Yeah, so I think it's in microarray. That's special in microarray.
Okay, so they assume that the microarray is accurate, so they want to adjust.
They assume they have some perfect or ideal microarray lie there, and they want to align other samples to that sample. So they have a baseline sample, in other words. I think in RNA-seq, you could also do the same thing. It's just that you need to pick one sample to be the baseline or target, and you just want to convert other samples to be like the sample. So that's a very common way of doing normalization.
Okay, I have another question. On this graph, the dots are genes. So
yes.
They're not. So this whole line is one sample, or.
Sorry, this x-axis is one sample to be normalized, and the y-axis is another sample you set as fixed. So you have one. So let's say you have 3 samples to normalize, then you will have 3 of these graphs.
I see.
Yeah, but the set H is the set, and so the number of dots here is the number of genes in this set H.
So the set H is the invariant genes, right?
Yeah, invariant genes. Yes.
So for the genes that are outside of this set H,
So for the set H is used for fitting this line, fitting this curve, and then you apply this curve to other genes.
So for another gene, it will be on the so this line basically like ordered by genomic position or anything. So if I have another gene, how would I How would I know how
Because
much?
it's a function, right? It's a function that can, like call it f hat. It can take any expression value and output it to some other
Okay.
value.
Okay. So okay, I see. Thank you.
Yeah. Right. So I think the reasoning is that we can only do this correction based on the set H, because genes in set H are believed to have the same expression value between the two samples. Other genes, they may not. Right. If every gene has the same expression in two samples, it doesn't make sense to do differential expression analysis at all. Yeah.
Dr. Lee, is this regression, the regression normalization, the same thing as the quantile normalization in your notes?
No, it's different.
Okay.
Yeah.
Thank you.
So we're going to talk about quantile normalization next. So, and then after that, we can revisit this one and we can discuss about how they're different. Okay, so quantile normalization, it is something that's known to be very useful in practice. And its idea is very, I would say it makes a lot of sense to me. It doesn't require to define an invariant set of genes. It's just based on the distribution of all genes within a sample. Okay. So the idea of quantile normalization is that if I look at, let's say, the genes in one sample, we get a distribution of their expression values. And then the quantile normalization idea is that we want to make that distribution the same for all the samples. So here the idea is that all samples have the same distribution of gene expression levels. So let's say that we have still the same notation and samples P genes. So before doing that, I want to introduce an idea which is called empirical distribution. That's a very important statistical concept. So let's say for sample I, okay, using my same notation, I would list its expression levels for the P genes as xi1 to xip, right? So I will have all those P values for P genes. And then I can define an empirical distribution. So what does an empirical distribution mean? Anyone who has learned it before and willing to speak? Yes.
I mean, it's basically you build the histogram, and then if you want to find the probability that the value is in between two numbers, you just take the number of values between those two numbers, divide that by your sample size, right?
Yeah, that's a very good explanation, very intuitive explanation for it. So to put it mathematically, we just give every value here, every one of the P values a probability mass. And that probability is 1 over T. So every point carries the probability. So in other words, just as Gabe said, it gives us basically a P value discrete distribution, because every value has the same probability 1 over P. And you can plot that distribution using a histogram, like in R, or you can do a density plot, empirical density estimation. That's another possibility. So you can plot that distribution. And so what does it mean is that, let's say, if I call this distribution F, usually in statistics, we call this big F, and we give it a hat because it's an empirical distribution, CDF, and the I stands for sample I. So basically it means that this empirical CDF, if I input a number there, X, it will give me its left tail probability. In other words, it's the number of the proportion of the P points that are less or equal than this X, okay? So I can just write it as j from 1 to p, and I use an indicator to check whether xij is less or equal than x. So please note that this indicator is very useful because it just gives us a binary output. If it's less or equal than, it gives us 1. Otherwise, it gives us 0. So basically, I'm using the sum and indicator and counting the number of the values among the p-values that are less or equal than my given input x and divided by the size p. Okay, so that's the empirical distribution. And given this empirical distribution, we can also talk about quantile. So quantile is another very important concept. So basically, the distribution and the quantile, they have a one-to-one relationship. So probably I can draw a figure here to show what it means. Okay, so for a distribution, I have two ways to plot it. Let's say this is x. I could either plot its density, probability density. So of course, here the density is actually this. So every dot here is 1 bar, right? So like the histogram is plotting that. So if the density looks like this, it means that a lot of these values are somewhere here. I have many values in the center and fewer values as I get far away from the center. So given this density, I could define, let's say this. So if this is x, then what I have on the left of it, this is what we call this fi hat x, the probability. And also inversely, if I give you a probability p, any value between 0 and 1, I can find its quantile by looking for, let's say p corresponds to maybe somewhere here, okay? So if I know the area to the left of it is p, then I could find this value, this corresponding value that give me a left tail probability p, and I can call this f hat I inverse p. So you know this is a function and this is the inverse of this function. So f hat I converts a value of x to a probability. And the quantile function converts a probability to a value in x. So this is what we call the empirical quantile. Okay. Another way to show the same thing is you can always draw the CDF, not just the density. So CDF, I can put it in the y-axis, and it goes from 0 to 1. That's what we define by a CDF. So you see that when the value of x is small, it starts from 0, and then it goes up to 1. And after it reaches 1, it stays as 1. Because it's just, when it's 1, it means your x values falls into this right region. That means the left tail probability remain as 1. When it's 0, it means that your x goes to the left region, which means that in its left, there's no possibility or probability of observing any values. So given this CDF plot, you can more clearly see that relationship between quantile and probability. So let's say that given p is right here, I go here and look for it, and this is exactly fi hat inverse p. I get a quantile. And for another example, if my x is here, I can go to here, and that will give me fi hat x. So there's a one-to-one correspondence between probability and the quantile. And that's the basic idea of quantile regression. So the idea is to make, so let's say that given this empirical distribution, how many quantiles do we have in total? P, right? So let's say that we store the values from the smallest to the largest. After the sorting, I could call this xi1 less or equal than xip. So we use this lower sub-index with parentheses to mean the sorted values. So after the sorting, I know this is the minimum, this is the maximum. And in the empirical distribution, you see that each of them correspond to one left tail probability. So this is the quantile of probability 1 over p. Okay, and or I could also write it as the one set as 1 over p times 100 percentile. You can say it either way. Or you can say it as 1 over p quantile. So these terms are all used. So correspondingly, this will be 100 percentile because its left tail probability is 1. Or you can say it's the 1 quantile. Yeah, probably this is not so good to say that, but yeah, you know what I mean. So this is the probability 1's corresponding quantile. So yeah, so the idea is that for every sample, after you do the sorting of the p genes, you can get p quantiles. And across the sample, we want to make sure that the quantiles are the same. That means my goal is to make all samples have the same 1 over p quantile, 2 over p quantile, and so on, up to the last one, the 100 percentile. So I want to make them all the same. Then what should I do? The idea is very simple. That is, I would take the first 1 over p quantile for all the n samples. That means I have n such quantiles. I would take the average of all of them. And then I would just use that average quantile to replace original quantile by changing the value in every sample. So in other words, I will do that for the 1 over p quantile, 2 over p quantile, up to the 100 percentile. And then all the values in every sample will be changed. So in other words, they will have the same p values for in each sample, but the rank is still the original rank. I will restore the rank. So to help you understand that, I'm going to give you a concrete example as in the notes. So I'll just simplify a little bit so you can see the idea. So let's say that in the example, we have, I'll simplify it by just having 3 genes, okay? Gene 1, 2, and 3. And I will still consider 3 samples, sample 1, sample 2, and sample 3. And I'll just make up the numbers over there. So sample 1, gene 1, I just put number 100, 80, 60, and 50, 90, 50. And then 30, 20, 30. Okay, so that's what I want to do. And then I'm going to keep the order in each sample. So I want to know what's the rank of each of the genes. So that I will change this to be, this is the expression matrix. Okay, what I'm going to do is to convert this to a rank matrix. And what I will do is that for the smallest value within each sample, I will call it rank 1. And the second is rank 2, and the largest is rank 3. So clearly you will see that here the rank gene 3 is rank 1, 2, and 3. And in sample 2, it's 1, 2, and 3. Sample 3, 1, 2, and 3. So I keep their rank. And then so basically here the rank 1 is corresponding to the first quantile. Here it will be 1 over 3, 1/3 quantile, and 2/3 quantile, and 100 percentile. I only have 3 quantiles to normalize. Okay, so then what I will do is that I will look at Here is the step called quantile normalization. I will make the quantiles to match with each other for each sample. So let's say that I would look at the rank 1, 2, and 3. So you can think of the rank as equivalent to quantile. So this will be 1/3, 2/3. And then I look at the sample. So what is rank 1 in sample 1? What is the quantile for rank 1 in sample 1? That's 30. Sample 2, that's 20. Sample 3, that's 30. So as I said, I want to match them. And the best way to match them is to take the average. So I take the mean. So that will give me 30, 20, 30. How much is that? So that will be 80 divided by 3. That will be the average across the 3 samples. For the 2/3 quantile, so in sample 1, it's 50. Sample 2, 80. Sample 3, 50. I want to calculate the average of those 3. So it's 180 divided by 3, that's 60. And finally, for the 100 percentile, in sample 1, it's 100. Sample 2 is 90. Sample 3 is 60. Okay, so after the averaging, it will become 250 over 3. So I got the average quantiles for those three ranks or 3 probabilities. And then I would input them back. So given the average quantile and the rank matrix, so together it will give me the new values. Okay, in the new values in the matrix, I still have gene 1, 2, and 3, and sample 1, 2, and 3. I will follow the rank and put their new values back. So for rank 1, the new value is 80 over 3. So that's for gene 3. Rank 2, it's 60. And rank 3, that's 250 over 3. So I'm done with sample 1. For sample 2, I put them back following the same original rank. So 80 over 3. The second rank is 2. So it's gene 1 that has rank 2. So that will give me 60. And it's 250 over 3. So the last one, I'll just do it directly. 60, 250 over 3. So you see what's changed. The value within each sample, they're all changed. They're all changed to the average quantiles, but you still keep the relative order of genes within that sample. Like gene 3 is still the lowest expressed, and the gene 1 is still the highest expressed, right? So you keep that order, but you change the actual values. And after doing that, you would make, if you plot the 3 genes values as a distribution, then each sample 1, 2, and 3, they will have the same distribution. That's what the quantile normalization is doing. Okay, so after introducing this technical procedure, do we have any questions? Then we are going to explain why this is making sense biologically.
Yes. Do you do quantile normalization after like RPKM and TPM?
Yeah, definitely. So all the normalization we're talking now are based on RPKM or TPM units. And usually the data you obtain are already in RPKM or TPM, unless you work with raw reads. Okay. So then why does it make sense to assume every sample has the same distribution across genes? The rationale is that we assume between biological samples, the majority of genes expression levels should stay as rather similar distribution. It's just that whenever we have some changes, it's a small number of genes whose expression go from low to high or go from high to low. But we believe that the overall distribution of expression levels across genes should stay rather constant. I think the reason for assuming that is for the analysis where your samples are serving sort of as replicates. So for that case, then those samples should have a similar distribution across genes. Do you think that's reasonable to you? Yeah, but I would say for single cell data, it may be a little different because we know that if your samples are cells, right, in single cell data, we know that some cells are bigger than other cells. For example, if you think about in blood, macrophages are bigger than red blood cells. So it's reasonable to assume that the total RNA amount is more in macrophages. So in that case, it's no longer reasonable to think, okay, within a macrophage cell, the gene expression distribution should be the same as the gene expression distribution in the red blood cell. So that's why I'm saying that normalization is a very subjective choice. It depends on what you believe to be invariant or unchanged before you do normalization. Okay, and so just as an introduction to the single cell case, since I already mentioned that, I'm going to share a screen with you with a paper which I think is quite interesting. So this is called as the norm. It's a work 2 years ago, I think, and published in Nature Methods. And it's about how we should do normalization in single cell RNA-seq data. And please note that your project data is single cell RNA-seq data. But one thing I need to say is that it's still debatable whether you need to do single cell normalization. I mean, if the cells are sequenced in one batch, let's say if you do blood cell sequencing, you sequence thousands or tens of thousands cells in one experiment. And essentially, you treat those cells in a very fair way, and you get the counts, right? Every gene will have a count in the cell. So it's debatable whether you need to do normalization on your data or not. So in my own group's research, we actually didn't do that when we have cells sequenced in the batch, especially if the cells are just, like the dropstick data. If the cells are labeled in a high throughput manner, and then you sequence it. But it's still, like I said, it's a very subjective choice. So like in this paper, they use a very interesting idea to do normalization. So let's look at this figure. So you can think of here, they group genes into 3 groups, okay? And so probably it's easier to think of them as 3 genes. Let's start from that. So this is for one, so this is bulk data. Maybe you can start from here. So this is bulk data. We have 3 genes, and each dot is a sample. Let's say if you have 200 samples, then you will have 200 dots here for one gene, 200 dots here for another gene, another 200 dots, because every gene has 200 measurements. And you are taking that gene expression level on the log scale, and you also take that sample's sequencing depth on the log scale. So you would imagine what? There shouldn't be any slope there. The reason is, you know, the gene expression should be linear in the sequencing depth, right? It should be sequencing depth times the relative proportion of that gene in the sample. And you know, the magic of log transformation is that you can make a multiplicative relationship as additive. So in other words, you would just have the log gene expression equal to log sequencing depth plus something. And the log sequencing depth should have a slope equal to what? It should not be, the slope should not be positive, right? Right, because you basically, you make that you made that multiplicative thing already in the intercept. So that's why they believe that there shouldn't be like a positive slope in those things, but they see that positive slope. So their idea actually came from this bulk data normalization. What they do is that they fit a regression line for each one. They do the adjustment based on the regression line, similar to what we said before. Then they make the slope equal to 0. Okay, so that's what they did for bulk. And they use the same idea for single cell. So in single cell, what they do is essentially the same thing. They think, okay, for this highly expressed gene, medianly expressed gene, and lowly expressed gene, this slope should be 0. And that's the basic idea of their SC norm. And they found that in single cell, sometimes the slope could be different for different genes. Like the red gene has a positive slope, but for the blue gene, the slope is close to 0. So they want to change this relationship to all these relationships to all have 0 slope for each gene. And of course, in the implementation stage, they did something a little trickier. They group genes together. So this is no longer one gene, but multiple genes. Because in single cell data, we know the data is very sparse. So you want to adjust for that. But I'm just pointing out that this is something good to know. And if you're interested, you can check out this paper yourself. Especially in this paper, they also mentioned some other normalization methods for your information. And also, like we're going to talk about dChip later. So in those methods now, they have their inherent normalization step for dealing with data. But of course, when you use the software, you could choose to skip the normalization or use it as a default. And one more thing I want to say is this method, probably I can show here. This is a very famous method for batch effect adjustment. It's called combat. I think it was originally developed for microarray data, but later the authors proposed versions for RNA-seq data as well. So let's take a look at this key figure. I'm also going to post it to Cecily for your detailed reading. So in this figure, you see that the idea of combat is based on generalized linear model, or specifically they do negative binomial regression. Maybe I can make this bigger. Okay, so in their notation, let's say that they use YGIJ to denote the expression level of gene G in sample J from batch I. So you know the sample index here is nested under the batch index I. So for example, let's think about single cell data. Then the batch I is the ith experiment, and the sample J would be the jth cell in that batch. So in generalized linear model, you basically need to assume 2 layers in your model. One is the distribution, how the counts are distributed around its mean. So here we assume the count follows a negative binomial distribution, which we'll also talk about later in this class. So please just take it as given for now. Negative binomial distribution with the mean parameter and the dispersion parameter. So you see that here, only the mean parameter has the J index because you assume that every gene G in sample J from batch I has its own mean, which is unknown, but we're going to assume it. And the dispersion parameter that only depends on the gene and the batch, but not a sample. And this is also very common. It's just that you assume the sample J's have the same dispersion. Okay, so for the mean, we use this log linear relationship. This is also typical in generalized linear model. So just for your information, I'm teaching a course called Stat 205 this quarter. It's specific about this linear model, generalized linear model. So if that's something of your interest, you may take that course later. We will not detail it in this course because itself is a whole course, but I'll just take it as something that's very commonly used, and I'll try to explain it in a very short, brief way here. So for the log mean, we assume that it's a linear relationship with an intercept, which is gene specific. but not sample specific or batch specific. And we have the sample here. So sample J is entered here because sometimes we have some covariate for that sample J. For example, the sample, you may have its library size, right? That's a very nice covariate to be considered. Or in other cases, for example, if the sample is human data, you may know the sample's owner or donors, age or gender. Those can also be used as the in the covariate vector xj. And we assume that there's a slope that is gene specific. So basically, the intercept and the slope are only related to gene, but not batch, not sample. And here, this is the effect we want to adjust for. There is a constant in addition to the common intercept. This one is batch specific. So in other words, we are wondering whether there is a shift in this log mu relationship that is just due to the batch I. So this gamma GI is undesirable. We want to remove it. Okay, and finally, we have this log NJ. I think it's here, it's the sample size. So basically, they didn't put the sample size here, but they leave it out here. So they are just arbitrarily assume the sample size has a log effect and the slope is 1. Okay, maybe I think they have some special consideration there. Okay, and this is a typical format just from the negative binomial distribution. So in the negative binomial distribution, the variance is equal to the mean plus mean squared times dispersion. So it's a known fact. So then what they do is that they try to fit this model, negative binomial regression model, for every gene G. So every gene gets its own model fitted. So once you have the model fitted, as we said, each parameter will have an estimate. So for the gamma GI, we will call the estimate gamma hat GI. And for the mean, we will have its, let me see, sample J. Right, so for the mean, we will have its estimate mu hat GIJ. And then we know this gamma GI should go away. What we did is to subtract this estimate from the estimated log mu. And we call this our new log mu GJ star. You see that now the batch I is gone. It's no longer here because we assume that after the normalization, there's no more batch. There is only sample and the gene. And that's basically it. So that's the idea of doing the normalization. So after you do that, you have the adjustment for every gene in sample J by removing the effect of batch I. And then we would just adjust the count accordingly. Because you see here, we will assume the adjusted count comes from this new mean and the new dispersions. I'm going to skip this part. It's not that important compared to the mean. So the mean adjustment is the key. And then you're going to draw your sample from this adjusted distribution. And you are going to keep the probability. So for example, if the original count corresponds to, say, left tail probability equal to 0.1 in this negative binomial, then you're going to get the same 0.1 quantile from this new NB, and that will be the new count. So basically, after that, your original count matrix will be adjusted to the new count matrix. So you can see that compared with the quantile regression or the quantile normalization we just talked about, we didn't have any specific assumption on how the data should be distributed like in our approach. And we are pooling genes together by assuming every sample has the same distribution across genes. But here, we are doing this adjustment gene by gene. So in this idea, we didn't use the across gene information, but I would say that it's just for this simplified version. In reality, when they estimate those parameters, they will use empirical Bayes, like we said in last lecture. And in empirical Bayes, you would assume those parameters to follow a common prior. That means different genes parameter, let's say for the same intercept, alpha G across genes, they will follow a same prior.
And by doing that, if you use empirical bias to estimate the prior parameters, you are essentially pooling gene information for the estimation. So this gene G's estimation will also be dependent on other genes. So I hope this makes a little sense to you for now. But I think the key idea is that we assume every gene has a specific NB, negative binomial distribution, in each sample and in each batch. And after the adjustment, we're going to remove the batch effect by only keeping the gene and sample effect. So this is a more complicated way of doing batch correction, but essentially batch correction is for the same purpose as normalization. You want to make the samples more comparable with each other. So does that answer your question? So for the quantile normalization part, do you have any questions? Okay. Okay, so I think we're good. So I'll post those two papers for your further reading if that's something of your interest. But again, like I said, I think normalization itself is such big topic, especially for people who work on normalization. They could develop variants constantly. So ultimately, from users' perspective, I think it would be very important that you really have the ability to understand what each method is assuming, and then pick the one whose assumption is making sense to you. Okay, so let's continue with our course. That's all I want to say for normalization for now. And our next topic is multiple testing correction. So we're going to bring it up here. And multiple testing is a very important thing to consider or do when you do high throughput biological data analysis. I'm still going to use the context of differentially expressed gene analysis as the example. So let's say that we have P genes, and we want to test whether each one is differentially expressed between two groups of samples or between two biological conditions. Then we will have P tests, right? Test whether each one is DE, differentially expressed. And I will list those null hypotheses as H01, H02, to H0P. So each null hypothesis is that the corresponding gene is not DE. And we're going to make a decision for each hypothesis, for each test. So why does it actually matter? We know that in hypothesis testing, a commonly used significance level is alpha, equals 0.05. So let's say for test I, right? We define the significance level we call it alpha as the probability of rejecting the null hypothesis I, given that the null hypothesis I is true. Although this null hypothesis being true or not is not something we can observe, but the significance level is something we target in our mind. We want to control this level to be small, so we do not make too many false rejections. So imagine that we set alpha equals 0.05. That's a commonly used level. Then it means that when the null hypothesis is true, we have 5% probability of mistakenly rejecting it, which means that we reject it with a false positive. Okay, so that means when H0I is true, we have a 5% probability of rejecting it. And that's what we call false positive. So when we're doing only one test, we're fine, right? Because 5% is considered to be a very small probability. But what if we're doing thousands of tests? So let's say that if we have 1,000 null hypotheses that are true, then how many false positives will we have? Then the expected number of false positives, would be 1,000 times that 5%, because each one has a 5% probability of being rejected. And let's say that we're assuming the simple case that each null hypothesis is rejected independently. Then this, actually, no, we don't need that because it's just a summation. Sorry, my bad, take it back. So each null hypothesis, which is true, has 5% probability of being mistakenly rejected. And we have 1,000 of those. So their false positive probability will accumulate, right? And then that's what? That will give me 50 false positives. So you can imagine that this is something that's not so good for us because the number of false positives will explode or will increase linearly as the number of null hypothesis increases. And in biology, we know that when we do differentially expression analysis, we believe most genes are not DE. Usually, our so-called DE genes will only be no more than 100. Otherwise, our analysis will not be so informative. So given that the majority of them are believed to be null, or the null hypotheses are believed to be true, then the number of false positives will be too big if we set alpha to 0.05 for every test. So this is the fundamental motivation of doing multiple testing correction. We want to adjust the alpha to be a much smaller level for rejection. So hopefully we can maintain this number of false positives to be quite small. Okay, so that's the basic idea of doing this hypothesis multiple testing correction. So before we do that, let's first introduce some notation. I think that's necessary as we have in the notes. So let's see that we have, so in the notes, we have M test. Probably let's do that later. So next class, we are going to introduce a notation for that matrix with a summary of the number of correct decisions, wrong decisions, false positive, false negatives. But for today, let's first define something that's quite conservative. It's called the family-wise error rate. Okay, let's first do that. And this definition doesn't have too much to do with the notation, FWER. So the family-wise error rate associated with the decision threshold, which is alpha here, is basically defined as the probability of making at least one false positive. Okay, so it means that we are trying to bound this family-wise error rate to be very small. So if we want the FWER to be less or equal than alpha. So let's say alpha is said to be 5%. If we want to control this to be very small, what should we do? So intuitively, you can think of this making at least one false positive as a union event. Let's say we have making P decisions, and we want to count whether each decision is a false positive or not. So when one occurs, the union occurs, right? So it's a union event. When one, if we make one false positive or two false positive or three false positive, it's all counted as this event, union event occurs. So we know that the very important inequality in probability, let's say that here, this FWER is just the probability of the union. Okay, J from 1 to P. That is the gene J is a false positive. If we make one gene as a false positive, we trigger the event. And we know that in probability, the probability of a union can be upper bounded by the summation of probabilities. Okay, so J from 1 to P, we can move it outside and just take the summation for the probability that gene J is a false positive. And because this event, when gene J is a false positive, means that the gene J is not EE, but we mistakenly reject the hypothesis. That happens with the probability equal to the significance level, right? So this is equal to summation J from 1 to P times the significance level, which we don't know how to set yet. So then you see that it's the same significance level times P, because the summation is for the same term. So it's P times the significance level. And then we want to solve for the significance level so that the FWER is under alpha. Then you can see it is sufficient to set the significance level to be equal to alpha divided by P. Right? Because if this is set to be alpha divided by P, we put it here, it's P times alpha divided by P. It gives us alpha. So then we can control the family-wise error rate under alpha, or 5%. So essentially, we are just adjusting the significance level for each test to be the family-wise error rate we want to achieve divided by the number of tests. And we'll call this simple procedure Bonferroni correction. So it is the most conservative way of reducing our false positives. So we are not looking at one test at a time, but we're looking at the P tests together. But you can clearly see that a disadvantage of the Bonferroni correction is that it's overly conservative. Like say if alpha is 5%, 0.05, if we divide it by 1,000, let's say P is 1,000, or usually it's better more than that, right? Greater than that. If you have 20,000 human genes, P is equal to 20,000. Then this significance level is super small. And if you compare each test p-value to this super small significance level, you may not reject any test. That means you will not call any gene as DE gene. So the drawback is overly conservative because alpha over P is very small. That's the issue. So Bonferroni was very widely used in the old days when the P is not that small. When people only do say 5 tests, 10 tests, or at most 20 tests together. It's not so much of an issue. But now with the high throughput experiment, we cannot afford to do this correction because maybe our interest is not to make any one false positive, but our interest is not to have too many false positives. So this motivated us to have something in between setting alpha to 0.05 and or to set the significance level to be much smaller than alpha, like out to 0.05 divided by 20,000. So that's the motivation for the new procedure called new criterion, I should say, false discovery rate. So it's defined as a new criterion for setting a threshold on p-values, not alpha over p, not alpha, but somewhere in between. So we're going to stop here for today. And for those of you, if you are interested in staying and having an after-class discussion, please let me know.